Hello everyone, in this video we are going to learn about transcription in detail, how that process happens, what is transcription in a detailed way. So in this chapter you will learn the sequence of events that take place in transcription of DNA to RNA and how those events differ in prokaryotic and eukaryotic systems, about the structures and functions of different classes of RNA. So this is a very important topic, but we will be dealing with this topic with the help of some more videos because it is difficult to include all this information in just one video. So we will be doing a lecture series. So first, transcription. Gene expression is accomplished by the transfer of genetic information from DNA to RNA molecules and then from RNA to protein molecules. RNA molecules are synthesized by using the base sequence of one strand of DNA as a template in a polymerization reaction that is catalyzed by enzymes called DNA-dependent RNA polymerases or simply RNA polymerases. The process by which RNA molecules are initiated, elongated and terminated is called transcription. The process by which RNA molecules are initiated, elongated and terminated is called transcription. So in this chapter we will quickly realize that although complementary base pairing is common to both RNA and DNA synthesis, several features of RNA synthesis differ from DNA synthesis. For example, the RNA product is single-stranded, unlike the double helix from which it is transcribed. It is also relatively short in length since only a small portion of the total DNA is represented in any single RNA product. Initially, we will learn about transcription in prokaryotes and we will review the more complicated eukaryotic gene expression system. Because of this relative simplicity, the pioneering experiments on transcription were carried out in prokaryotes. In fact, it was a series of elegant genetic experiments with E. coli that led to the discovery of what is now known as messenger RNA or mRNA. Through mRNA, the information stored in DNA is converted to amino acid sequences of the ultimate gene product, the protein. Three aspects of prokaryotic transcription will be considered in this chapter: the enzymology of RNA synthesis, the signals that determine at which locations on a DNA molecule transcription starts and stops, and the types of transcription products and how they are converted to the RNA molecules needed by the cell. So which are the three aspects? The enzymology of RNA synthesis, the signals that determine at which locations on a DNA molecule transcription starts and stops, and the types of transcription products and how they are converted to the RNA molecules needed by the cell. Then we will examine eukaryotic transcription and finish the chapter with a review of a laboratory procedure for studying RNA. So first, enzymatic synthesis of RNA. I have told this is going to be the first topic, enzymatic synthesis of RNA. So in this section we describe the basic features of the polymerization of RNA, the identity of the precursors, the nature of the template, the properties of the polymerizing enzyme and the mechanisms of initiation, elongation and termination of synthesis of an RNA chain. The essential chemical characteristics of the synthesis of RNA are the following: like the precursors in the synthesis of RNA are the 4 ribonucleotides, 5' triphosphates like RNTPs, ATP, GTP, CTP and UTP. On the ribose portion of each NTP, there are 2 OH groups, one each on the 2' and 3' carbon atoms. 3' carbon atoms. In the polymerization reaction, the 3' OH terminus of the RNA being synthesized reacts with the 5' triphosphate of a precursor RNTP. A pyrophosphate is released and a phosphodiester bond results. This is the same reaction that occurs in the synthesis of DNA. The sequence of base in an RNA molecule is determined by the base sequence of the DNA. Each base added to the growing end of the RNA chain is chosen by its ability to base pair with the RNA strand used as a template. Thus, the bases C, T, G, and A in a DNA strand cause C, G, A, C, and U respectively to appear in the newly synthesized RNA molecule. But the DNA molecule is double-stranded, we know that. Yet at the same time, any particular region is transcribed, only one strand serves as a template. This comes from because the two DNA strands are separated temporarily to allow one to be used as a template. The RNA chain grows in the 5' to 3' direction, that is, nucleotides are added only to the 3' OH end of the growing chain. This is same as the direction of chain growth in DNA synthesis. Also, the RNA strand and the DNA template strand are anti-parallel to one another. So, mechanism of the chain elongation reaction catalyzed by RNA polymerase, the pyrophosphate group and the hydrogen atom do not appear in the RNA strand. The DNA template and the RNA strands are anti-parallel as in double-stranded DNA. So 5' triphosphate and B base region, then base, nitrogen base, 5' triphosphate region, phosphatase, phosphate, etc. are very important. So an RNA strand is copied only from strand B of a segment of a DNA molecule. No RNA is copied from strand A. Strand A in the sense some strand which is not copied. Okay. The another one which acts as a template strand that is being copied. However, elsewhere, for example, in a different gene, strand A might be copied. So we are assuming two kind of strands in RNA, like strand A and strand B. Strand A acts as a template in one cell, but in another cell there is strand B as a template. However, elsewhere, for example, in a different gene, strand A might be copied. In that case, strand B would not be copied in that region of the DNA. The RNA molecule is anti-parallel to the DNA strand being copied and is terminated by a 5' triphosphate at the non-growing end. RNA polymerase, in contrast to DNA polymerase, are able to initiate chain growth directly. That is, no primer is needed. Only ribonucleotide 5' triphosphate participate in RNA synthesis, and the first base to be held down in the initiation event retains its triphosphate. Its 3' OH group is the point of attachment for the subsequent nucleotide. Thus, the 5' end of a growing RNA molecule terminates with a triphosphate. The overall polymerization reaction will be written as NNTP plus XTP DNA RNA P Mg2 plus, like XTP NMP raised to N plus NPPI, in which XTP represents the first nucleotide at the 5' terminus of the RNA chain. NMP is a mononucleotide in the RNA chain. E. coli RNA polymerase consists of 5 subunits, 2 identical alpha subunits and one each of types beta, beta dash and sigma. The sigma subunit dissociates from the enzyme especially, and in fact does so shortly after polymerization is initiated. The term holoenzyme is used to describe the complete enzyme and core enzyme for the enzyme that lacks the sigma subunit. We use the name RNA polymerase when the holoenzyme is meant. RNA polymerase is one of the largest enzymes known and can easily be seen by electron microscopy. So the synthesis of RNA consists of 4 discrete stages: binding of RNA polymerase to a template at a specific site, initiation, chain elongation and chain termination and release. A discussion of the stages follows. So one thing to remember, RNA polymerase, in contrast to DNA polymerase, are able to initiate chain growth directly. That is, no primer is needed. Remember that. Then transcription signals. The first step in transcription is binding of RNA polymerase to a DNA molecule. Binding occurs at particular sites called promoters, which are specific sequences of about 40 base pairs at which several interactions occur. The most crucial interaction for positioning of E. coli RNA polymerase at a promoter occurs at two short sequence patches on the DNA. These patches are located about 10 and 35 base pair before the first base, which is copied into RNA. That first base copied is called the start portion of transcription and is assigned as position +1. The two patches therefore are at positions 10 and 35 or negative 10 and negative 35 with respect to the start point. The sigma subunit of RNA polymerase appears to have two separate alpha helical segments with amino acid residues that can make specific contacts with the exposed portions of the base pairs in the major grooves of DNA. The enzyme becomes positioned correctly when the separate alpha helical segments contact the base pairs in the two patches simultaneously. Since a DNA molecule makes one helical turn every 10 to 11 base pairs, these contacts would be nearly on the same side of DNA 2 turns away or 78 angstrom along the DNA. Extended RNA polymerase could contact those two patches. E. coli RNA polymerase is a large protein that converts as much as 70 to 75 base pairs of DNA from position 55 to position +20 when it is bound to a promoter. It can therefore easily make the two separate contacts at the same time. The sequence of many promoters have been identified by researchers. Portions of the sequence along one strand for a few E. coli promoters with the start points and negative 35 and negative 10 regions are very important. The strand that is shown at its 5' end at the left, one striking feature is that no two sequences are identical or even closely similar except in the two key locations. The sequences are -7 and -12 are similar, as are the sequences from about -30 to about -35. The sequence around -10 are considered to be variants of a basic sequence TATA80 called the -10 census sequence, while the sequence around -35 is a variant of TTGACA called the 35 census sequence. A consensus sequence is a pattern of bases from which actual sequences observed in many different systems differ by usually no more than one or two bases. Although the feature only shows the sequence in one strand, remember that the DNA is double-stranded and that the other complementary strand is part of the recognition site in the -10 and -35 regions. With some promoters, the sequence at one of the two contact regions differ greatly from the census. In this case, RNA polymerase recognizes the sequence very poorly and the promoter does not function very well. For some genes, that might be fine because the cell may not need to express that gene very often. For other genes, however, there is often an accessory protein that helps RNA polymerase bind to the promoter. This kind of accessory protein is called a gene activator protein and basically is a positive effector. For example, the lambda pre-promoter is active only when the lambda C11 protein is present. These gene activator proteins usually bind to specific sequences very near or even within a promoter sequence, and they appear to have surfaces to which RNA polymerase can attach when correctly positioned on the promoter. This gene activator protein helps the RNA polymerase bind to a promoter with a poor recognition signal. An important effector protein is very important and is the CAP protein. That protein is needed for the activation of many promoters for genes required for sugar metabolism, and regulation of the binding ability of CAP is a major means of regulating the expression of these genes. A diagram showing RNA polymerase bound to DNA at a promoter. The arrows indicate the position. I'm sorry for mentioning that. Then CAP is a major means of regulating the expression of these genes. Then after RNA polymerase binds tightly at the promoter region, it manages to unwind a small portion of the DNA from base pairs +3 to -10 to form the open promoter complex. This unwinding is necessary for pairing of the incoming ribonucleotide triphosphate, the building blocks for synthesis of RNA. The base composition between the start site through the -10 region is generally rich in 8+T, which renders the DNA especially susceptible to unpairing. Once an open promoter complex is formed, RNA polymerase is ready to initiate synthesis. RNA polymerase contains two nucleotide binding sites called initiation site and the elongation sites respectively. The initiation sites prefers to bind purine nucleotide triphosphate, namely ATP and GTP, and one of these is usually the first nucleotide in the chain. Therefore, the first base on the DNA template that is transcribed is usually a thymine or a cytosine. The initiating nucleotide triphosphate binds to the enzyme in the open promoter complex and forms a hydrogen bond with the complementary DNA base. The elongation site is then filled with the nucleoside triphosphate that is released strictly to its ability to form a hydrogen bond with the next base in the DNA strand. The two nucleotides are then joined together by a reaction. Let's continue this session with the help of next video.